This method can help answer key questions in the endocrinology field as well as the neuroscience field, such as the research to reveal brain responses to endocrine signals.
The main advantage of this technique is that you can examine direct effects of endocrine signals on the brain separated from other tissues.
To begin this procedure, use forceps to scratch the bottom center of a plastic culture dish to create a dent for easier mounting of the ventral ganglion of the larval brain after dissection. With a toothpick, place a small drop of superglue on either side of the dent and attach a tungsten rod to the glue. Then, using a small piece of putty-like reusable adhesive, make a circular wall surrounding the dent. To dissect the larval brain, 90 to 120 hours after egg laying, collect larvae and wash them at least 3 times with distilled water to remove adherent food debris. Next, place a larva on a square 1.5-inch watch glass filled with ice-cold PBS. Use a pair of forceps to gently grab the middle part of the larva. Use another pair of forceps to grab and pull the mouth hooks gently to separate the anterior part of the larva containing the brain from the rest of the body. Then, hold the anterior tip with the forceps and turn the larva inside out.